Enough. Okay, good. Well, thank you all for coming today. It's Ed's confirmation seminar today. Ed's a Sydney boy like myself. There's not many of us around, so it's nice to have another one here. He did his undergraduate degree in here at James Cook University, over at CEAS, which I think has ceased to exist, what used to be TSAG. Yeah. And he's had a fairly diverse background, Ed. It'll take a while to get stuck back into his studies, but he spent time at the Great Barrier Reef Marine Park Authority, where he is an accomplished aquarist, and he's also a superb photographer as well, so he brings a number of skills to his PhD. Anyway, so now I'll pass over to Ed to talk to you about it.
Thank you, Andrew. All right. So what I'm going to talk to you about is coral depth zonation, and this is an important aspect of coral reef ecology because it's something which is a really important part of how corals distribute, not only over space but over depth, and it's something that I think we should pay a little bit more attention to these days. It seems to have dropped a bit by the wayside. So what I'm going to do is I'll tell you a little bit about the introduction, situate my research, the aims and research questions that I'm going to look at, the research objectives, so the depth zonation of coral assemblages, the depth abundance distribution of species, the traits which are characteristic of species with these differing depth distribution abundances, and then finally the population performance over depth and what this really means for all of the distributions that we see. And then some expected outcomes at the end of this project. So we'll get started, and when we think about the basics of ecology, it's really about the study of the processes influencing the distribution and abundance of organisms. It's all about where things are and why they're there. So one of the basic concepts is that species aren't distributed evenly in space or in time, and understanding how these patterns are caused and maintained is a real major goal of ecology. When we think about coral reefs, these are hyper-diverse ecosystems. They dominate the tropical shallow oceans. And Wells noted in 1957, nowhere else in the seas is there such a bewildering range of living things, and also perhaps nowhere else is the pattern so uniform, characteristic and widespread as in the coral reef. So he's talking about the zonation of corals. He's talking about it over biogeographic scale, so among reefs and between reefs. But one of the more prevalent of these patterns in zonation is depth zonation in corals. It occurs over a much shorter scale, but it's a very strong gradient, and it's something which is really prevalent in coral reef ecosystems. So the depth diversity relationship that we see in corals, describing this was really done between 1960 and about 1990, and there's four of the main studies which looked at this. So I'll go through what they found and the sort of patterns that we're looking at. So Grow and Wells started in 1967, which is this yellow line here, and they looked at the number of species that they saw at each depth at which they sampled, and they saw reasonably low species richness in the shallows, and then a decline from about 5 metres. In 1972, Yossi Loya went to Eilat in the Red Sea, and he looked at evenness. So this was on a transect scale on the relative abundance of each species within that transect. This was a diversity measure, and he went from 0 metres down to 30, and he saw an increase, a consistent increase all the way up to that maximum depth in diversity. In 1980, Charles Shepherd went to the Chagos Archipelago, and he looked at species densities. This is the number of species that you see on any given sampling unit, which was a transect in this case. And this is this blue line. Again, low species density in the shallows, but again, low species density in the deeper water as well. So it tails off beyond about 30 metres. And then Kuhlman in 1983 used the range size of each species and put these all together and asked how many species could inhabit at each depth which he looked at. And this is this green line on the outside. And again, low in the shallows, peaking around 40 metres, but then drops off towards 60. So the thing that all of these patterns have in common, despite the fact they're all using different metrics, and they're over different years in different locations, what they all have is this low diversity in the shallow regions and low diversity in the deeper regions. And somewhere in the middle, we've got a hump. And this was kind of taken as a pattern that we see in coral reefs. We see this nice hump-shaped pattern, and this is what we really thought was happening with diversity. And the reason this was taken so much at face value is the fact that we see this in so many other ecosystems. So when we look at birds over altitude, for instance, this is Carsten Rybeck, we looked at it in 97, and found, again, low species richness in the low altitudes, and in the high altitudes, again, low species richness, and then a hump-shaped pattern in the middle. So this was a consistent pattern we're seeing across different ecosystems, and this is really taken as what we're going to see on coral reefs. And we try and explain this pattern. Houston tried to do this in 1985, pulled all these data together, and looked at the things which were describing how this distribution was coming out. And there are a couple of main environmental variables contributing to this. And the first is light. So light is really important for a lot of reef-building corals. They require on photosynthetic symbionts for a lot of their energy. So as light decreases exponentially with depth, we see a decrease in the amount of productivity that you can get over this depth range, the amount of energy being acquired by corals. What we also see is these different gradients. So wave energy is a great example. This is a disturbance gradient. And in the shallow water, we're seeing a high frequency as well as a high intensity of disturbance. And as we go down the slope, it gets consistently more and more stable in deeper water. So we get increasing disturbance in the shallow water, decreasing productivity in deeper water. And the way that these actually built these patterns was based on Connell's idea of the intermediate disturbance hypothesis in 1978. So although these shallow regions have high productivity, they also have such high disturbance that you get a lowered number of species coexisting here. It's a fringe habitat and not many can actually reside and thrive in these regions. Conversely, in the deeper habitats, we've got a very stable environment, but the productivity is so low that not many species can actually establish and then grow. And a lot of the growth down here is so low that things like competitive exclusion, which can operate in shallow waters when there's no disturbance gradient, they slow down to actually become relatively non-existent in these deeper regions, which can explain a bit of a lift in the species diversity with depth. But this is sort of where we left it. There's a few issues with these studies. The first is that there's been some major revisions in coral taxonomy, particularly over the last decade, and this isn't so much of an issue for a lot of studies, but it's important here because a lot of this data was collected between 1960 and about 1990, so it's a fairly restricted time range, and it leaves it open to a lot of problems associated with this taxonomy. So what we were previously describing as taxonomic units or species may not actually be what we now see, and this may affect the patterns that we're looking at. There was also inconsistent sampling methodology. You can see that in some cases, this is Porter in 72 and Professor Baird just last year, a lot of things haven't changed, but some things have. So Cornell and Carlson tried to pull all this data together in 2000 and looked at all of these things. They tried to pull the data together and look at the way that this hump-shaped pattern was created and how consistent it was. And they found that it was really hard to do this because you had things like line intercept transects, unconstrained counts where people just swam around collecting corals, vertical transects up the slope. We also had varying sized quadrats all over the reef. So there was a real disparity in the way that this data was collected. It never really got together. There was also little data from deeper sites. It's quite hard to sample down deep, and this is becoming increasingly problematic in recent years. So there's a lot of effort put towards shallow regions and successively less put towards increasing depth. And all of these studies also tended to use different metrics to describe diversity. So even species richness, species evenness, and species density, which we saw in the previous slide, all of these were used, and they were used in similar ways. And they all do describe diversity, but they describe slightly different aspects of diversity. So it may not really be a consistent way to look at the pattern that we see. And this can also influence what we're actually finding. And when we talk about the way species distribute over depth, one of the first things that you notice is depth ranges, and this is used for a lot of studies. This represents the best data that we currently have. This is from Carpenter et al. In 2008, and I've compiled about 200 species from this data set, and it's displayed on the x-axis here is just each species consecutively ranked by the median depth at which they occur. And each white bar represents the maximum and minimum depth range of one species and the space that they occupy. And the first thing that you notice is that there's these rather blocked patterns in the data. So the reason why that occurs is that this isn't really empirical measures, this is expert opinion. So we get a lot of people in the room and say, who's seen this coral where? And someone says, oh, around 20 metres. And it's not really an empirical estimate, and you tend to end up with these consistent blocking patterns. And it's a bit of an effect of lacking data. So what I did is, in the start of the year, I went up to Kimby Bay, and just over a month I sampled a whole bunch of colonies and the extent at which they covered. And this is represented in this green data on the bottom. So I've used exactly the same display over exactly the same species, and where these are extending is where what I've recorded extends beyond what's actually in the literature. And the first thing that becomes fairly apparent is that there's a lot of range extension, particularly in the deeper regions, also some in the shallow regions, but there's a significant undersampling of coral ranges below 15 to 20 metres. And this is going to have an effect on how this data actually looks. And that's quite important because this is used for a lot of things. It's the basis of a lot of key studies which are being done these days, really high-profile studies. Things like Keith et al. We looked at biogeographic patterns, but the best data that we have for looking at range sizes is this, and range sizes came out as a significant factor. Again, Carpenter et al. Actually derived this data to look at the vulnerability of each species to future extinction, and one of the main features was the range over which a species can occur. So these are significant findings, but the data that we've actually got is probably undersampling a lot of these deeper ranges. So I need to start to look at this again and really build on what we've got. So this brings me to the research gaps, and there's a few. The first is that the depth zonation is really inadequately described so far. We've got to start and I want to keep going. The shape of the diversity gradient is still unclear, and the structure of the assemblage that actually builds this is relatively unknown. Species are also assumed to occur pretty much evenly across their entire depth range. An examination of within-range patterns is really lacking, and the traits that are characteristic of these different depth distributions haven't been explored either. And trait-based ecology is something which has come on in leaps and bounds in recent years. And finally, the population-level performance of a species, so how it fills this niche which it occupies, is really still quite unknown. And the aims and objectives of my study, so how I'm going to actually come up with a way to look at this is, firstly, I'm going to look at the structure that exists, or what structure exists in the depth zonation of coral assemblages. Secondly, do species actually specialise over depth? Can we describe how species use depth, their depth preference and their specialisation? Thirdly, what life history traits characterise the depth distribution of a species? And fourthly, how does performance of a species alter over depth? So I'm going to start by defining the pattern, move down to the next level, which is looking at the specialisation extent, and then the traits which are characteristic of these extents, and finally the population performance and how they use this niche. So we'll start with the depth zonation of coral assemblages and the diversity pattern that we see. So I want to ask, how does this hump-shaped, or does this hump-shaped pattern actually exist? And I'm going to do this by describing the changes in diversity with depth, and I'm going to use a consistent sampling methodology. And I'm also going to use a couple of different types of data analysis to try and tease apart where these different results are coming from. After that, I'm then going to look at the structure of this pattern. So whether this is a random pattern or a nested pattern, there's a whole number of ways that an assemblage can actually be structured within this nestedness, so I'll look at that a bit later on. So my study site, I'll be going to Kimby Bay in Papua New Guinea for a couple of reasons. First of all, it's got a really nice depth profile, so I can get a really nice sample range all the way within the bay. It's also within the Indo-Australian archipelago, so this is the diversity hotspot of reef corals. So we'll get a lot of species distributing over a really nice range, and we can look at them quite clearly. I'm going to sample at 3 locations, inner bay, mid bay and outer bay locations, with 3 reef sites within each location. And at each location I'm going to sample from 0 to 40 metres over this reasonably wide range of depths. So the methodology I'm going to use, because current methodology really fails for time for a lot of these studies, are very time restricted when working at depth. Things like line intercept transects and quadrats can't really be applied when I'm working below 20 metres. And things that get around this time constraint, like ROVs, AUVs, video transects, don't often have the resolution that I'm looking for, things like species level identification, and they also tend to be financially crippling to actually run. So I'm proposing a new sampling methodology, and this is going to be based on a technique which is established for bird diversity surveys in terrestrial ecology. It's been quite well tested, and it's going to encompass these time restrictions that I've got, quite strict time restrictions at depth, require minimal equipment, so I won't be messing around with tapes, minimal habitat requirements, so I won't need a continuous section of reef at a particular profile in order to complete this survey. Importantly, it has to be replicable at any site, so I can actually get out and use this and create a consistent method. And it has to have a high detection rate of rare species. And this is important when I'm looking at diversity questions, because I really want to actually encompass the diversity I'm seeing within a site. So what I'm going to do, so I'm going to find my reef profile and run a vertical transect from 0 to 40 metres. And at each 5 metre bathymetric interval, I'll have 1 count station. So at each of these count stations, I'll have a 5-minute time limit, and I'll count 12 colonies, which has been established as the most consistent number I can actually identify within that 5-minute time frame. Each colony will be sampled from a central colony at the decided depth along the transect, and then consecutively outwards to the next nearest colony from the previously sampled one up until 12. They'll all be at least 5 centimetres in diameter, so I can get a proper ID on them. and I'll record the ID to species level. Where I'm not confident of an in-water species level ID, I'll take a high resolution image. I'll record the depth to the nearest 10 centimetres, the colony width to the nearest centimetre, colony length to the nearest centimetre, and I'll also take into account the growth position of a colony. So whether it's living in a cave or in a cryptic environment, because this can confound a lot of these results that we're getting on depth distributions. I want to take that into account. I've actually gone out and tested this methodology, and I've done this using species accumulation curves. So what this actually represents, this is a test of this new method, and this is the observed species as they accumulate on increasing numbers of samples, and showing the species richness which results from this. This represents a rarefaction curve, so this is the estimate of this curve extending forward, the predicted species count with a 95% confidence interval surrounding it. And finally, this little line up here is a total non-parametric richness estimator. So this takes the number of rare and incidental species that are being added along the way and extrapolates this out to try and predict how many species I would eventually find if I sampled every single colony within a certain region. And the important thing about this is that what I'm seeing is that around 300 individuals sampled, I'm seeing a bit over 80 species recorded. And if I extrapolate that out and I look at a doubling in the sampling effort, I'm only getting about 20 more species recorded. So I'm capturing most of the species diversity within those first 300 samples. And this means that these extrapolations are much more constrained, and it means that any results I'm going to compare at this level are going to be much more accurate. The other thing is that this estimated curve is starting to hit its asymptotic level and actually narrow down. And the confidence intervals that surround this at low sampling are very, very wide, but at this range they're really narrowing down, so we're getting a much more precise answer. And this means that I can also compare this across sites as well as the species accumulation level at a similar sample. What I also did is went out and ran a consistent method, a 10-metre line intercept transect. A lot of coral reef ecology is based around this method. And I ran this over exactly the same site as this new methodology. And what I found was that while this is really good for picking up the assemblage of the assemblage that occurs at any reef, what I'm seeing is that the number of species that are being picked up is far lower. It's not picking up these rare and incidental species as well. And also this total richness estimate is actually below the observed species count at that site. So we're seeing a significant underestimation of diversity using this method, which means that this one that I'm proposing is far more robust towards this. I also compared the assemblage structure that I was seeing between the line intercept transect and what I was getting on this count method, and they came out quite similar, which is very promising because this is a very well-established method for looking at these kind of community assemblages. So back to Kimby Bay, what I'm going to do is look at these three locations over the depth range, and I'm going to apply 12 replicate vertical transects at each reef site. And this is going to mean around 140 colonies per depth per reef, and importantly over 400 colonies per depth per location. And this is the region at which I'm going to start comparing this data. And around 1,300 colonies per depth for the entire bay, and over 10,300 colonies for the entire bay. So this is quite an extensive data set, and it's going to get the resolution that I require to do this analysis, which will be looking at this pattern. So describing the depth diversity pattern that exists in corals, I'll start by looking at the species range overlap. And this is what Kuhlmann did in 1983. And you take all of the species range extents, put them all together and ask how many of these extents is encompassed at each depth at which I'm sampling. And I've done this using the preliminary data that I've collected from Kimby Bay over this range. And what we see is that nice reminiscent hump-shaped pattern. This is quite similar to what we've seen over altitude with birds, and it's similar to what's already been established. But if I then take the species accumulation curves, and these control a lot for effort, and I apply that to the same data at around 200 samples sampling effort, so a bit less than what I'd expect to see on the final data, this pattern all of a sudden starts to disappear and there's no real change over depth. So I'm seeing a significant effect not only of the sampling methodology, but also of the way that we're actually analysing these results. And this can contribute a lot to the way this pattern is described. I'm also going to describe it using the richness estimation, but the current data isn't a high enough sample size to start doing that yet. Secondly, what the structure of this pattern is. And the way I'm going to approach this is I'm going to use an elements of meta-community structure analysis. Now this essentially takes this graph we've seen before. This is what I displayed the Carpenter data in, with increasing species over increasing median depth, and then the range they occupy. And this is using the actual data I've recorded from Kimby Bay. It's much more clean data. And what I'm going to ask is essentially to turn this into a matrix and then ask how this matrix is constructed using this technique. This was established by Liebold Michelson in 2002, and it has been used by Presley et al. In 2012, Keith et al. In 2013. So this is a fairly well-established methodology. And I'll look for 6 idealised patterns within this structure. So a random, checkerboard, Clementsian, Gleasonian, nestedness structure, or an evenly spaced structure. And I'm going to do that using 3 elements. So the coherence, the species replacement over that range, as well as the species range boundary clumping. So how many species are ending at exactly the same depth. So once we've actually looked at that in a bit in depth, we'll look at the next level down. So the depth abundance distributions of a species within its range. And the thing is that species are often described as depth generalists and specialists based on their depth range. We can say if it occurs over a wide range, it's a generalist. A smaller range tends to indicate a specialist. But species aren't distributed evenly in space. We know this. This is a basic concept of ecology. We know it's true for species over a depth gradient. We've just seen that. So why would it be true for species over a depth range? And what I want to know is how do species actually specialise over depth? And not only specialise, but where their distribution is actually deployed. So to define the depth specialisation and preference of a species, I'm going to use the depth range, but I'm also going to use the relative abundance over depth for each of those species to give a little more data to this. And the reason this is kind of important is I'll show you, this is again using data from Kimby Bay over 3 species, a Merulina, a Parietes and a Pachyceras. And if we use it in the classic one-dimensional frame, everything occurs between 0 and 40 metres. They've all got exactly the same distribution. They're all exactly the same type of coral. They all use the same depth range. But if we look at the way that they're distributed over depth, we see a different depth preference emerging from this specialisation. So although the Merulina tends to prefer the shallower water, the Parietes, if anything, sort of gets a bit everywhere. And the Pachyceras particularly likes to seem to like these deeper regions. So the depth preference is another way of describing this specialisation level of a species. And you can add an extra element to this. And the way I'm going to try and describe this is using a beta function, which has been developed by Josh Maiden down in Macquarie University. And what this does, it describes this pattern using 3 parameters. And the parameters are the depth range over which it occurs, between 0 and the maximum, the depth specialisation, so the shape of this curve, or the acusity of the peak, and finally the depth preference, so where this peak actually occurs in space. And these 3 parameters will be fed into this model. And what it will essentially describe is the distribution of a species on these 2 axes, alpha and beta. And the way that this works is that values with a high beta and a low alpha will tend to describe shallow specialists. High alpha and a low beta will describe a deep specialist. High values in both will be a mid-depth specialist, and low values in both will tend to be a generalist. So we've got generalists here and specialists tending to be further out along these axes. And if I take those 3 example species and ask, well, in a normal analysis that all occur over exactly the same point, so where would I expect to see them in this distribution? And what I'd expect to see is that while they all occur within this generalised area, they're actually distributing laterally along this depth preference. So I'm getting another level of information out of this data. And it's another way to start to describe how these species are using depth. So then we'll move on to the traits. So the traits that are characteristics of species with these contrasting depth abundance distributions. And what's defining these? So life history theory proposes that these certain suites of these traits are favoured in particular habitats, i.e. deep habitats and shallow habitats, and the species that occur within them. So organisms, for example, that reproduce early in life should really be favoured in environments where disturbance is frequent, and this will be reflected in the traits. And there's been suggestions raised about the different reproductive methods, so brooders and spawners, and how these actually change over the depth gradient. Stimson in 1978 suggested that brooders were predominantly shallow due to this disturbance effect. But the empirical data we're getting doesn't really match this. If anything, we're seeing brooders down deeper. And this has significant implications for other ways of looking at coral reef ecology, particularly things like the Debrief Refugio hypothesis. And this is the idea that deeper sets of species can actually rebuild shallow water communities that have been damaged. But Bongartz in 2010 looked at this along the traits that were within these 2 deep and shallow environments, and found that depth may not provide this refugio because he was seeing different suites of life history traits between the two areas. So we might not be getting that interchange. And this is why this sort of question is quite important. So the aims of this study is really to ask, do species with differing depth abundance distributions, which you've already seen, do they have a different suite of life history traits within them? So there's a few significant traits that I want to look at. For example, egg size. So we'd expect to see deep specialists having shorter gametogenic cycles. This is a product of reduced energy acquisition, so we'd see that trait more predominant in deeper water corals, for instance. Polyp size. There's the idea that deep specialists will have a larger polyp size. Again, there's lower energy acquisition for photosynthesis down there, so we'd expect to see a higher level of heterotrophy, so maybe a larger polyp size in deeper corals. And the analysis I'm going to use to try and get at this is a multiple linear regression approach. So the dependent variable of this is going to be the depth abundance distribution, and this is going to be an axis defined by the beta function, which I've used in the previous chapter, and over 200, probably over 300 species that I'll actually accumulate from the data in this initial sampling run. And the independent variables are going to be a number of traits which will be derived from the CoralTraits.org database, which has recently become available. And for example, things like egg size, polyp size, the mode of larval deployment, brooders or spawners, and the sexuality of these species these are going to be example traits which will actually help describe the biology of these species and possibly be important when describing their depth distributions. So the traits that significantly explain the distribution along these axes are then going to be identified. We can start to look at how well they partition over depth. And finally, I'm going to look at the population performance over depth, so how species actually use this depth which they occupy. And population performance it's usually measured by intrinsic population growth, so this is the growth rate of a species at a particular location that it's being measured. And there's several demographic traits that we can use to measure this. Things like growth, fecundity, mortality, and recruitment are all ways we can inform this. And what is really necessary is integrating all of these units to inform the actual population level dynamics of a species over its range. And I want to ask, can population performance actually explain the contrasting depth abundance distributions that I see? Again, I'm going to go back to Kimby Bay. It's a nice site to work with on this depth range, and I'm going to take two species identified with differing depth distribution abundances within this first section of this study. I'm going to look at 3 different reef sites, and at each site I'm going to look at 4 10-metre blocks of depth. So 20 colonies per species will be tagged at each of these depths, at each of these reef sites. And these will be revisited after 1 year to look at a single year cycle in all of these demographic features that I'm going to actually record. And these will be, firstly, growth. And the way I'm going to look at growth is through an image analysis. I'm going to look at the change in volume of each colony over that 1 year interval. I'm going to look at the fecundity through a value of eggs per polyp, and this will be from fragments dissected from each colony, which will be then expanded to a value of polyps per centimetre squared to look at a size effect, and finally come up with a value of eggs per colony. This will be a size-specific effect, so the amount of eggs produced for a colony of a specific size at a specific depth. Thirdly, mortality, and again I'll use the image analysis used for growth, and I'll look at the total mortality of corals, so whether it's still there or whether it's entirely dead. And fourthly, recruitment. The way I'm going to get recruitment is by using a size structure analysis. I'm going to do this using that really detailed data that I've collected in the first part of this study to get at the recruitment level and the size structure of these assemblages. So the analysis for this, this is still a work in progress, but I'm going to look at these demographics by trying to use an integrated projection model. Now, this was done by Josh Maiden in 2012, and what he did is he took this model to combine a lot of theorised or model parameters, particularly around energy acquisition and also exposure to hydrodynamic gradients. And they looked across the top of a reef flat in distance away from the reef crest. And I want to turn that sideways, and then I'm also going to use a lot more empirical values instead of model parameters to hopefully simplify this, and also to try and come up with a more unified way of looking at how these species actually change. So the expected outcomes from all of this, what am I going to see? Well, hopefully I'll see a number of papers come out of it, and all of these will actually be things that I expect to see produced regardless of what the results actually are. These are all going to be publishable units, regardless of what I'm actually seeing in the data. Hopefully we'll also be able to attend a couple of conferences and actually report this to people. ACRS in 2015 and 2017, and the ICRS in 2016, when a lot of this data will actually be out, and I'll be able to present it to people. I have got a timeline, so we're over here. A lot's being done, and the next 2 years I've got enough space to do all of my data analysis, all of my data collection, hopefully publish along the way, which will leave me my last year free to actually deal with my thesis and write everything up and get everything done within the time frame that I've got. I do have a budget. Total budget's looking at around 21,900. Clearly this is going to change quite a bit. Funding source is going to be primarily through my supervisors. I'm an AIMS JCU student, so I can also apply for these research training grants. Hopefully I'll use one of these to get over to a macroecology lab in Denmark and start to work with a few of the guys who really know their stuff, particularly for the statistical work. And I'm also going to be applying for several grants, things like the Ecological Society of Australia and the International Society for Reef Studies along the way. So thank you for all coming to staying, and if you've got any questions, please let me know.
I'll ask one.
Yes.
Ed, you're focusing in Kimby Bay.
Yes.
One of the early hypotheses that you put forward was the intermediate disturbance, saying wave action is greater in shallows, etc. Kimby's pretty benign.
It is quite benign.
So how do you think that may influence?
Well, this intermediate disturbance hypothesis works in a slightly different level to things like the randomness or the structure of the community assemblage. So what I'd expect to see in a differing energy environment is to see maybe this hump pushing further up the slope, or maybe I see the way that this distribution would then mimic the disturbance effect that I'm seeing in Kimby Bay. So if I'm getting a structure which looks like it's going to come along those lines, I can also analyse things like the wave energy at each depth and the light attenuation and these disturbance principles. And you're right, there are some particular quirks to this environment. It's got a very different kind of wave matrix and reef matrix to what we see on the GBR, and it's got a slightly different set of parameters when it comes to disturbance. So yeah, something to bear in mind.
It's not always bathtub either.
That's true, yeah.
And there is a gradient, and you're sampling across the gradient.
Yeah, but hopefully we'll find a consistency within places that have the same sort of wave energy. Yes?
Very good talk, thank you. It was very interesting. So I work with conservation, and it was very interesting. You started your talk by kind of presenting carpets very keys, and saying that it was nonsense, right?
Well, I think it works good, but
that was good though. And you really caught my attention when you said that, and then you went into a very interesting discourse on how you're going to actually collect more accurate data in very different ways, and I thought that was exciting.
Yeah.
But then I was slightly disappointed at the end because you didn't kind of like link it back to maybe why you were doing this work, right? So are you going to, my question is, are you going to link what you're doing to revising what Carpenter and Keith?
Oh yes, that's a really good point, and it's something which will inevitably be the end result of all of this work.
Yeah.
So if we start to rebuild things also like the Debrief Refugio hypothesis, which I can get at through the traits database. So I'll have the tools available to actually start to get into these again. Exactly how I can do it, I think, will depend on the results I'm getting and the sort of analysis I'm doing. But yeah, dead right. I think that's the end result of a lot of this work, is to try and revisit and to build on some of these studies that have these implications for conservation and management.
What I'm insinuating also is that if you do actually link it back to the red list, because they're actually red listing corals, then it'll be a very powerful tool to make more accurate predictions.
Yeah, hopefully. I mean, the better data we get, the better science we can do.
Yeah, there's a paper about it in one of the papers was about that.
Was it?
It wasn't mentioned specifically.
Just to add to that conversation, one of the motivators for this project is exactly what you're saying. But to get there, you got to do all this stuff.
Sure.
So yeah, we're hoping this will be the building blocks, which people can then start to add on along into the future. I think it's exciting. Hopefully. Yes.
I'm interested in when you were talking about identifying your 12 corals in 5 minutes.
Yeah.
What you think is the proportion of colonies that you're going to be able to identify underwater versus with photos, and whether you have any idea of, if you take more photos, do you end up with more species?
Yeah, that is something that, this is the reason we did this sampling at the start of the year, to try and look at things like that, because it is really important. Taxonomy is a bit of a basket case at times. But what I found is I was getting probably about half to 2/3 of the species, I was getting ideas that I was fairly confident with underwater, and if they weren't exactly the species that I was matching to, at least they were conforming to a taxonomic unit. So if I had these species, and I will have images along the way, hopefully I can then go back and work with some of the taxonomists and maybe put a name to it, but I want to make sure that I'm talking about the same thing. But yeah, the more images you take, the less certain a lot of these become, but it's kind of a trade-off. The more time I spend in the water, the more consistent it gets, the less images get taken, and the more consistent a lot of these IDs will actually get. So I came back with, after a month in Kimby developing this technique and getting some preliminary data, I came back with just short of 4,000 colonies actually in water ID'd, and these have all gone down to be identified now, and it's coming out quite well, and I'm quite confident that what I see will actually represent a fairly non-biased way of looking at these things. But there's also, hopefully we'll get a couple of taxonomists from MTQ up to Kimby as well, and point at some of the weird things and say, what the hell is that? And there's a lot of things. I mean, within this data set I've got, there's a significant portion which just can't really identify. I know they're a separate unit, but no one can really put their finger on what they are. So hopefully we'll start to also add to this information, particularly in the deeper water acroporoids. It's just a huge basket of no one really knows, which we need to sort out, hopefully.
Is there any plans in the future to, I mean, you specifically identified Kimby Bay, to then go through one or two sites in maybe the northern GBR and sort of analyse whether there's any bias just to Kimby Bay and methods, or it's replicable, the results are the same in other reefs? Yeah,
exactly. It'd be a really good thing to do. What I'm trying to get is somewhere where I can deploy this in different locations, in different places all over the world, and hopefully build data which is actually comparable, so we can start to look at these biogeographic changes as well. But yeah, there's a couple of things I'd like to do. I'd like to expand the range size of this so we can ask things along a three-dimensional gradient, not just depth, but also along these biogeographic distributions. And as well as that, I also want to start to push deeper. We're seeing that the biggest change in community structure is occurring somewhere around 60 metres. So this range will give a really good idea of what the actual pattern is, but if you want to fill in those blank areas, you need to start to really sort of inform those deeper regions, as well as building outwards as well. So the more places I can apply, I think the better this gets. But as long as the methodology is consistent, and hopefully, because this can be deployed in 5 minutes, anywhere at any depth, we should be able to just consistently build and build and build on this. But yeah, love to. Ideally. Yeah.
Just got a quick question. When you talked in the kind of the preliminary results where you showed how a lot of the coral species were occurring deeper than previously thought, and you said that's most likely due to the fact that it was expert opinion.
Yeah.
Do you think, is there any potential that these corals over that time period between when they were initially sampled and now could have been moving deeper, or?
Well, it's something that we haven't really considered, this temporal shift. I haven't even really thought about that. It's a good point. If we're still in a flux change, what we're seeing is, between the 70s and now, we've obviously seen big changes in coral reefs worldwide. So we might already see the development of these changes in distributions and changes in structures. It's something that we might be able to look for a signal in the previous data. But the fact that the data is so blocked and a lot of it is unknown, it's very hard to actually pull that apart.
Right.
But yeah, better records, I think. It'll be a really interesting thing to look at, whether there's a temporal component to how they're actually using space. Any others?
Okay.
That was easy. Thank you.
Thank you.